Baoga. I'm fine, sir. Good afternoon, sir. Yes, sir.
So in
Who are you trying to meet?
good afternoon, everyone.
Good afternoon, sir.
My name is Haruna Isiakumwa. I'm the founder of Computer-Aided Therapeutic Discovery and Design Platform, Federal University of Technology, Akure. This is a training I wanted to organize for all volunteers that are working with us at CATDD. So at least it should be part of the training related to visualization of molecular documentation, metabolism, excretion, and toxicity. want to pay attention to the as oriented, and you should take note of what steps our presenters are doing. Also, we as every oh, full of noise. How is it now?
It's much better now. Thank you.
Okay. Okay. So yeah. So after each presenter, there will be question and answer session, and we hope to cover a lot of ground today because I have a very hectic international course I attended, so I've been very been down. So that's why I want some other people to take you through the course of molecular docking. And this is a free training. Please use it well. It's a very rare opportunity because after this training, the subsequent one will come with a fee. So for now, I will give the floor to the first speaker, which is Victor Omoboyede, discussing and putting us through ligand and protein preparation for molecular docking. So, Victor, you on the floor.
Okay. Good afternoon, everyone. Please, can we hear me? Okay. Thank you. So, my name is Zomabuye Victor. So, I'll be taking us through the process of the preliminary process of molecular docking. So, I'll be sharing my screen now so that we can see my slide coming. I'll be sharing my screen now.
Hello, Victor. I want to ask you a question.
Okay, sir. I can hear you, sir.
Can we record this session?
Yes, I'm doing that, sir.
Can we record this session?
Yes, I'm already doing that, sir.
Good, please. Thank you.
Yes, sir.
Thank you very much. Then you can make a screenshot of the screen. We need to put it on the LinkedIn page.
Okay, sir. Okay, sir.
Yeah.
I'll do that, sir.
Thank you very much. Yeah. Thank you very much.
Okay, I'll be sharing my screen now so that we can proceed with the training for today. I'm coming, please. Can we see my screen? Hello.
Yes, we can.
Okay. So once again, good afternoon to us all. My name is Omobuye Victor, and I'll be taking us through the preliminary process of molecular docking. So just to give us a brief overview of what molecular docking is, I won't be dwelling much on this due to the limit of our time, but I'll be giving us a brief overview of what molecular docking is. So I'm sure almost every one of us here will be familiar with the concept of lock and key theory that is commonly used in the study of enzymes. Like it is a common theory that is commonly taught in enzyme. So just like that key and lock and key that is used to explain the concept of the catalyzation of chemical reaction. So molecular docking is also a technique for investigating the interactions between two molecules at the atomic level. Now, when our lock and key interact, it is not just it is not just an interaction. The interaction occurs at the atomic level. That is, there are some atoms that are responsible for the interaction that is observed. Let's say we take paracetamol now, and maybe the paracetamol is acting on the cyclooxygenase 2 enzyme now. So the kind of interaction that the paracetamol will have when on that with the cyclooxygenase 2 can be studied using molecular docking. So when we want to investigate the kind of interaction that our small molecule or any kind of molecule we have with another molecule, that is when molecular docking is employed. So the goal of molecular docking is to determine if two molecules will interact favorably and the best orientation for interaction. So now, not all molecules are interact favorably. We are still going to be understanding this in the course of the lecture. So molecular docking helps to determine, okay, if two molecules, you have a protein, maybe a protein or a protein ligand, if they will interact favorably and the best orientation of the protein. We all know that proteins, let me say, effector proteins or enzymatic proteins, they have an active site. So um, this active site is where we are going to be docking against. So the orientation of the ligand in that active site is what we are aim to study using molecular docking. So there are several types of docking. There are two classes based on the type of site that we are docking against and based on the type of molecule we are docking against. So I'll be focusing on the one based on the type of molecule we are docking against. We have the protein docking, which is commonly used in maybe in vaccine design and some other techniques like that. So we have protein docking, whereby you are docking two protein against each other, like maybe your vaccine and a toll-like receptor. You want to check if the vaccine can stimulate the toll-like receptor. We also have protein ligand docking, which is very common. So for protein ligand docking, you have your protein, which can be maybe cyclooxygenase 2, cyclooxygenase 3, and some other enzymes like that, and a ligand, maybe quercetin or paracetamol as well, and some other common ligands that we are familiar with. So protein ligand docking is carried out between a protein and a ligand, which can also be a library of ligands. So moving on, in cheminformatics, ligands are referred to as small molecules that are able to bind to and modulate the function of a protein. So now, in ligands, in coordination chemistry now, what is defined as ligands is a bit different to what we are referring to here. So ligands are just small molecules that can bind to and modulate the function of a protein, like the paracetamol that I said earlier. When we take in paracetamol does not just move around our body. Paracetamol has a target. So, and when it binds to that target, it is able to modulate, that is, it is able to control the function of that target. It's either so the it either serves as an agonist or an antagonist in the active site of a protein. That is, okay, when we take paracetamol and it binds to to its target, let's say COX2 now, when it gets into the active site of that enzyme, it can either act as an agonist, that is, it stimulates the protein to perform its function, or an antagonist, that is, it blocks the natural ligand of that protein. Yeah, like let's take for instance insulin and insulin receptor now. So insulin receptor, when insulin is secreted, what it will bind to is the insulin receptor. So, but when let's say we have a small molecule now that is able to bind to the active site of insulin receptor. So that when insulin is coming to bind to insulin receptor, the small molecule is already present on the active site, so it will not be able to perform its function. So that is an antagonist. While antagonist, it will stimulate insulin receptor to perform its function. I hope we all get that. So the small molecules they can either be natural or artificial compounds, and sometimes both. Like now, natural compounds like quercetin, curcumin, and all other compounds that I'm sure we are very familiar with, and that are mostly used when it comes to molecular docking. or phytocompounds, they are natural compounds. While we also have artificial compounds, like our paracetamol is an artificial compound, and some other compounds that are synthesized in the laboratory like that. So they can either be natural or artificial compound and both. So now, moving on, ligands to be used for molecular docking. How do we get the ligands that we are going to use for molecular docking? We can get them from literature. Like now, as I was saying earlier, I said we can either have a small molecule to be a natural compound or an artificial compound. So if a small molecule is an artificial compound now, if it is an artificial let's say I have my own laboratory now, so I synthesize a compound, then I test its activity against a particular protein. So such protein, such small molecule, you may not be able to download it from PubChem. You may not be able to download it from PubChem, but you will have it on the paper I published about the compounds that were synthesized. So you will have it there. But there are many databases that holds the structural details of ligands, be it synthesized one that have been approved or non-approved one as well, or other natural compounds. There are databases for them. There are databases for them. So in molecular docking, what is required is the structure. So sometimes you may have the smile, that is the smile representation of the ligand you want to work with, or you may have the smile, or you may have the maybe the PubChem ID or the Chembu ID and some other ID like that. That is not what is required. What is very important is, you can hear me. Hello, can everyone hear me? Network. And then also your slide okay. I was not okay. Sorry, I had issues with my network. Hello, can you hear me?
Yes, we can.
Oh, I'm so sorry for what happened earlier. I had a little connection issue. So I'll just recap a few things that I have said. Kindly give me a few seconds. Okay, now I said in cheminformatics, ligands are referred to as small molecules that are able to bind to and modulate the function of a protein. So now if we have a protein and we have maybe a natural compound that can bind to the active site of that protein and like change its function, maybe either inhibit or activate its function, those are referred to as small molecules and they are ligands. So they can either be natural compounds or artificial compounds. Artificial compounds, they can be synthesized in the laboratory, while natural compounds you can get them by from plant and some other from plant and some other things like that. So they can either serve as an agonist or antagonist in the active site of a protein. Like I said earlier, if we were able to get that, when a compound, let's say paracetamol for example again, when we take paracetamol has its own target, which is most times cyclooxygenase 2. So when paracetamol is taken into the body, it does not just move around, it goes for its target, which is the cyclooxygenase 2. And what it does to cyclooxygenase 2 is to inhibit it. So when it inhibits it, it is serving as an antagonist because the function that cyclooxygenase 2 is supposed to perform, it will not be able to perform it. But when we have a natural compound that binds to the active site so as to stimulate the action of that protein, that is an agonist. So moving on, so ligands to be used for molecular docking can be sourced from many places, including literature and databases, among others. So now we can retrieve the structures of our ligand from databases. We can also get them from literatures. So as I was saying, I can synthesize a small molecule in my laboratory. So when I synthesize it in my laboratory, its effect has not been, although I have tested its effect in my laboratory, but the effect has not been widely tested. So you may not really have it on databases like that. That does not mean my small molecule is not a good one, but it has not been widely recognized. It has not been deposited on databases yet. So you may not have, if you want to work with such ligand, you will not have them on databases, but you will have them on the literature or on the paper that I published after synthesizing the molecule. So ligands, you can either retrieve their structure from databases, or you can get them from literature. Like now, let's say you want to get the compound, the phytocompound that are present in Mangifera indica now. You may not be able to get everything with a search keyword on PubChem or any other database. But if you search literature, you can easily get the compound. Okay, maybe some people have done some maybe GCMS analysis on the extract or any other thing. Then they get some particular compound, they publish a paper on it. So you can easily get those compounds that are present in Mangifera indica from literature. So what is required for molecular docking is the structure of the ligand, not the smile, not the PubChem ID or any other form of ID. The structure is what is required for molecular docking. And there are databases to download the structure. We have the PubChem, we have Zinc, we have Chembu, we have Drug Bank, and many other databases like that. So PubChem is the most commonly used. PubChem is the most commonly used. Why like for zinc, any compound you see on zinc, it is commercially available. That is, if you need it, you can easily order it. Maybe you want to do some experimental work, you need to get the compound, you can easily order it. So let's and so to retrieve phytocompound as well. If you want to retrieve, okay, let's say you are working with allium cepa now. You want to get the compound that are present in allium cepa, which is onion. There are databases that hold information about the phytocompounds present in a particular plant. So example of such database is the IMPACT server. That is IMPPAT. IMPACT server, Indian Medicinal Plant and Phytochemistry Therapeutic Database. So if you can easily search, okay, I'm interested in Mangifera indica, you can easily search instead of going for literature. You can easily search okay, Mangifera indica. These are the compounds that are present in Mangifera indica. Then you can easily get their PubChem ID from that database, then get their structure from PubChem. So you are still going to go back to PubChem or any other database to get the structure of the compounds, because that is what is required in molecular docking. But in the case of if you are trying to retrieve phytocompounds now, phytocompounds present in a particular plant, to get the information in a place rather than just searching for papers here and there, you can just go to the impact server that I mentioned earlier. You are going to have them there, but you have to go back to a database to get the structures of those compounds. So moving on without wasting much of our time, you can also obtain the structures of ligands by drawing. So now let's say, like I was saying earlier, I synthesized a molecule in my a small molecule in my lab. You don't have it on any database. You need the structure to do some work with it. So how do you have the structure? Then you can draw it using maybe Marvin Sketch, Chem Sketch, and some other tools that are used for drawing. So you can easily draw. It's not something hard, so we can easily watch a few tutorials on it. 10 minutes, 15-minute tutorial, and we are good to go. So we can get the structure of our compounds by drawing. So and when we draw, we are going to save it as Mo2 file, which can then be converted to a SDF file, which is the structure data file. So if you retrieve the structure of a compound from PubChem, you are going to have it as SDF file. So when you draw, you are going to have it as a Mo2 file, which can be converted to an SDF file. So all other formats should be converted to SDF for using PyRx software. So it is very important. It's something that we should do. Okay, let's say you draw a particular compound, you have it in Mo2. You should convert it to SDF, maybe using OpenBabel. OpenBabel is a common software that is used for file conversion in drug design and discovery. So you can just easily convert. You have it in Mo2, Mo3, or any other format. You can easily convert it using OpenBabel or Avogadro. So now moving on now to proteins or target selection in molecular docking. So we already discussed what ligands are and how to get their structures. Now let's talk about protein structures. So protein selection is a very important part of molecular docking because that is what you are evaluating your compound against. So this is because selecting a bad protein can lead to problems during other downstream processes. Like now, if you select a bad protein, you will have issue in molecular dynamics. Now, some things that are worthy of noting when we are selecting protein. The first is we should avoid selecting protein with missing residues. And how do we recognize proteins with missing residues? I'll be showing us that when we quickly move to the practical session very soon. So, and if available, select a protein with a cognate ligand. So, what is a cognate ligand? They are also called co-crystallized ligand that are used to define the active site of a protein. So, I'll be showing us that. So, it's just that we now during the elucidation of the 3D structure of proteins now, they may use NMR, X-ray crystallography, cryo electron microscopy, and so on. But to define the active site of that particular ligand, so that the active site can be known to everyone, they will just they will just co-crystallize a ligand, which are referred to as cognate ligand, with it. So they use the cognate ligand to define the active site. So when you want to do your molecular docking, you can easily use some softwares to get to define the active site based on the co-crystallized ligand. Also, proteins with higher degree of resolution are good for docking. I'll be showing us this. So by that I mean higher degree of resolution here does not mean uh, resolution score does not mean maybe 4 or 5. Those with maybe 1, 2, maybe maximum of 2.5 if it is available are very good for docking. But when you are having it greater than 3, the resolution is as the score increases, the resolution the degree of resolution decreases. As the resolution score increases, the degree of resolution decreases. I will be showing us that when we go to the protein data bank. So we should know that we should avoid selection of protein with missing residues. We can, if available, we should select a protein that has a co-crystallized ligand, and we should select protein with good resolution scores. So in case what you have on PDB is a protein that has some missing residues, you can easily model that protein. You can easily model the missing residues using software like Modela, but Modela is paid, so you can just use a Swiss model server. It will model the incomplete residues of that protein, then you can then proceed to use it for docking. So now protein slash target selection as well. Now you may be interested in working with a protein whose structure is not on the protein data bank. It happens very well. So how do you go about it? You can get the structure of your protein by homology modeling. So the tertiary structure is not available on PDB. It's not available on a protein data bank. But the primary structure, which is the amino acid sequence of that particular protein, is will likely be available. It will likely be available. Is it better now? Hi, Ivana. Sorry for the delay. It's a little network issue. Hi, Ivana.
It's okay now.
Okay, thank you, sir. So if we are working with a protein that does not have its experimental structure, is the structure that was experimentally elucidated on the protein data bank, we can obtain that structure by homology modeling using the popular Swiss model. So you get the primary structure, which is the amino acid sequence. You get it from UniProt. Then you can now take it to databases like you can take it to servers like Swiss Model, RaptorX, and so on. But it is advisable to use AlphaFold because AlphaFold was recently recognized as the best software, I should say now. Let me just say the best predictor of 3D structures of protein. That is what I'm trying to say is if you have the premise sequence, the premise structure of your protein of interest, if you have it on Uniprot, you can easily use the Uniprot ID of that particular protein to retrieve your protein, the predicted protein on alpha food. So why is this advisable? This is advisable because the alpha food has the best prediction result. Currently, alpha food is the best, and what they have done is they have predicted they have predicted the 3D structures of every protein that you have on UniProt. So just like RCSB, which hold the 3D structures of protein, UniProt holds the primary structure, which is the amino acid sequences of protein. So what AlphaFold did was they predicted the structure of everything that we have on UniProt. So instead of you doing homology modeling or any other type of modeling, you just get the UniProt ID, go to AlphaFold database, then you retrieve your structure. Or you go to UniProt, you just click on structures. You are going to see AlphaFold. That is the structure that was predicted using AlphaFold. Then you can easily retrieve your protein instead of doing homology modeling. So what AlphaFold will give you, if you evaluate it, like using maybe the Ramachandran plot or the Z-score analysis, you are going to get a better result compared to Swiss model, RaptorX, and others. So when you obtain the structure from AlphaFold, then you can further process it, such as maybe structure refinement and evaluation, which I'm going to. Okay, for structure refinement, I will just talk about that later. So protein preparation. The first thing is to understand the structure of the protein. Are there missing residues that you need to model? If there are no missing residues, you remove non-amino acid residues. That is maybe the co-crystallized ligand, water. Now, if you download the structure of a protein, you are going to. It is during the process of elucidating the experimental structure, you are going to have water in it. So you have to remove water or any non-amino acid residues. That's it. You are interested in the amino acid residues that make up a protein. You are not interested in the water molecules and other molecules that are non-amino acid. Then you add hydrogens and charges. You minimize the energy of the protein. Then you save the protein in the PDBQT format. I'm going to show us all this very soon, so I won't be dwelling much on it. Now moving on, preparing ligands for docking. We have ligand preparation in molecular docking. Is the slide okay now?
It's not.
Okay,
I
I'll
can
just
see.
go with this. Now talking about preparing ligands for docking. So it's just we all know the concept of garbage in, garbage out. That is, if you give whatever you supply is what you are going to get. So just it also applies in docking. If you start with a bad structure, you are going to get bad result at the end of the day. So the result you are going to get at the end of the day will not be the perfect result. So ligand preparation is usually performed to obtain the best starting pools for docking, and the steps involved are the conversion of 2D to 3D. Most times you are going to have the 3D structure available on PubChem, so it is advisable to download that. But in case you drill the structure is going to be saved in 2D. So it's very good to convert the 2D structure to 3D, because in the 2D structure the hydrogen atoms will not be added. But when you convert it to 3D, you are going to have the hydrogen atoms added, just like we talked about in protein preparation, that we are going to add hydrogens and charges. Same thing applies in ligand preparation as well. Same thing applies in ligand preparation. You add hydrogens. So when you add hydrogens, so you are going to know if your small molecule can form hydrogen bond interaction with your protein, because you are going to also add hydrogen in the protein preparation process as well. So you add hydrogen. After adding hydrogen, then you energy minimize. So for the energy minimization, we use a phosphate. For energy minimization, we have the universal phosphate, the GAFF, which is general amber phosphate, and some other phosphates like that. So for the energy minimization, I'm going to be showing us better in the practical session very soon. So after energy minimization, then we are going to save our result. We are going to save the structure in PDBQT format as well. So the ligand preparation often gives rise to more reliable docking results. So if you compare the scores of a ligand that was minimized, that was prepared, if you compare the scores of the one that was prepared to the one that was not prepared, you are going to see the differences. And also, you can have false positive results when you don't prepare your ligand. That is, you predict the interaction that you are going to have when you don't prepare your ligand. If you prepare the ligand, you are going to have a different interaction. We can try that to see. So, energy minimization involves the use of force field. We have the molecular mechanics, the semi-empirical, the density functional theory, and so on. So now, the molecular mechanics is what we mostly use when it comes to molecular docking of the time. Like if you are working with 1,000 molecules now and you start doing density functional theory for each molecule, that is going to take your time. But the accuracy of the minimization increases downward. That is, molecular mechanics density functional theory is very, very accurate and much more accurate than the semi-empirical and the molecular mechanics. However, that does not mean the result that you get from the molecular mechanics is not perfect. So I'll be illustrating that without wasting much of our time. I've taken much time already. So I'll be illustrating that now. Sorry, I'm coming. So now I will quickly retrieve the structure of a compound from PubChem. And to navigate to PubChem, you can just Google search PubChem, PUB and CHEM. When you Google search PubChem, the first result is going to be the link to the database. So I will be retrieving the structure of quercetin. So what we have the search bar here. What we are just going to do is to search quercetin. Quercetin is a natural compound. Quercetin is a natural compound, and we have the structure of quercetin here. We have the smice here. Now this smice, we are going to be needing it, so we can just copy it and paste it somewhere. But we are not going to be needing it for my own part. So for quercetin, you just click on the name. When you click on the name, it will take you to the page that you can download the structure. Also, I'll be going to protein data bank to retrieve the structure of a protein. So to go to protein data bank, you can just search protein data bank on Google. We can just search protein data bank. The network is a bit slow. So now we have the result for protein data bank. The first result is also the link to the website. The first result is the link to the website. So here we have the page for quercetin. I'm sorry, my network is crawling. So here we have the page for quercetin, and to download the structure, on the right side we are going to see a download icon and the word download. So we have the 2D structure and we have the 3D conformer. We just save the 3D conformer. We click on save, and when we click on save, we are going to have the structure here, which we can rename to fit the compound name. So also to retrieve a protein from a data to retrieve a protein now from the protein data bank, which is RCSB. So when the site loads, we are going to have an interface like this, and we have a search. We have a search. We have a search bar. So let's say I'm interested in working with P53 now. I can just search okay P53. That's human P53. And the reason why this is showing this particular icon that I'm hovering right here is because I have PyMon installed on my system. So if you don't have PyMon installed on your system, we just you will have this blank. You have only the name and not this particular icon that we can see right here. So PyMo is a tool for visualization, so we can just download it. You can download it. So now, I said earlier that structures with we should consider structures with lower resolution score when we are selecting structure. So to see that, if we click on sort by score, then we can say, okay, resolution from best to worst. If we say resolution from best to worst, we will see that it's going to rank the ones with the lowest, as we can see right here. This one with a score of 0.48 is the best. Then it's going to be increasing downward 0.6. It's increase downward and downward like that. So it is very advisable that we just go for the ones. We should not go for something more than 2.5 most times, something more than 2.5. But we should just be cautious when we are selecting our protein. The ones with the lower resolution scores are the best. The ones with the lower resolution scores are the best. So I'll be going back so that I can properly search for the P53 that I'm interested in. So I'm interested in P53. Then for Homo sapiens, Homo sapiens P53. Okay, now we have some. This is 2.1, and this is 1.92. This is 1.92. Comment, please. I'm trying to refine the search term now. I'm trying to restrict it to those of Homo sapiens alone. So we can say here that the name has changed, the organism name has changed. We have it as Homo sapiens already. So for this as well, we have it this as a 1.35, which is the crystal structure of HDMX bound to P53 peptidomimetic and some other information like that. But we should just scroll down so that we can get the best structure. From the name, we can easily know that okay, this fits into the scope of my search and this does not fit into the scope of my search. The structure of human MDMX, that is not relevant to our search then. I'm sorry for the delay. Okay, this is 2.5, although this is quite high. This structure is quite high, but we can just open it in a new tab to inspect it later on. This structure is quite high. You can just scroll down to get other P53 structures. I'm sorry, when I refine the search term, it mixes up the result. It mixes up the result. I'm going to rectify that now. Okay, I've opened this everywhere. Just search for another protein. I'm sorry for the mix up. This is for most musculus. I'm looking for that of Homo sapiens. Crystal structure of EGFR complexity. This is quite high. 3.14 is too much. Okay, now I was talking about missing residues earlier. So when we see something like this in the structure of a protein, I hope we can all see that. So when we see something like this, these lines that are right here, these are broken lines. They represent missing amino acid. They represent missing amino acid residues. So selecting these is a very bad thing. So we can see the resolution score as well that it is 3.25, which is very high. So this is not a good structure. So this is what I meant by avoiding proteins with an incomplete amino acid residues. But if we want to select this, we can easily model the missing amino acid residues using maybe a server like Swiss model, but you may be questioned because there will surely be better structures available than this. So if you have to model the incomplete residues, then why don't you go for the ones that have completed residues, including this as well? This has a missing residues. Okay, EGFR complexity. We'll just go for, although the resolution is 2.5, but I'll just go for it so as to save our time. So now this one has a resolution of 2.2. So let's go for this particular protein, this cis-D-UK. The cis-D-UK. Let's go for it. Better than the ones that we've seen so far. So now to download, okay, just some information before we download the structure. We have the method. The method for the elucidation of the structure is the X-ray diffraction method. But when we scroll down, this is the literature that comes with it. We should reference the literature whenever we retrieve a particular structure. So the literature that comes with it should be referenced. But this is what I'm much more interested in. So now what this is telling us is that five it has 5 chains. It has this particular protein that we are about to retrieve has 5 chains, which are chain A, chain B, C, D, E, and F. So in the process of our protein preparation, we have to there's something that we should take note. We should know if it is a homodimer or an heterodimer. So if it is a heterodimer, we have to keep them intact. If it is an heterodimer, then we have to know what kind of heterodimer is it. Are some chains different? Like let's say like in hemoglobin now, we have two alpha, two beta. So the two alpha and the two beta, if they are the same, there's no point in using all the 4 chains for molecular docking. But if they are different, then we can then okay, let's retain the 4 chains and use it for molecular docking. So in most cases, we are going to be having homodimer. So when we have homodimer like that, we should just remove one of the chains and use the other chain for molecular docking. But when we have heterodimer, we should use heterodimer, then we can now do more research. Okay, what kind of heterodimer is it? Are all the chains the same or are they all different? When they are the same, you just use one, and when they are different, you use everything. So this has given us a kind of information about the protein that we are working with. Then this is telling us that these are the ligands that have been used to define the active site, especially these particular ligand that we have here. This that we have here, we can easily use this to define amino acid the amino acid residues in the active site of this particular protein. So to download the protein, we click on download files, then we click on PDB format. So we click on PDB format. Now going for protein preparation now, we use a software called Chimera to prepare our protein. We use a software called Chimera. We can easily search Chimera on Google and we are going to be able to download it. So I will open the protein using Chimera now. I will open the protein using Chimera. I want to share this screen.
Hello, Victor. You've even overused your time.
Well, I'll soon be done, sir. In about
No,
five,
no problem, Sha.
6 minutes, I'll be done.
It seems people are enjoying your flow.
Thank you, sir. I'll soon be done. So what we are going to do now is we are going to open it using Chimera. So then we can select the chain that we are going to. Then we can the co-crystallized ligand as well. This is the Chimera software now that is currently loading. Then we can separate the co-crystallized ligand. We can separate it from the protein. And when we separate it, we are going to save it in a separate format because we are going to use it to validate our docking. So when we see something like docking validation, the co-crystallized ligand is what they use to validate the docking. But just to quickly show us what I meant by incomplete residues again, once more. This particular protein that I have on my screen now, it contains incomplete residues. That is why we have these particular broken lines here. So and we can see that Chimera is already giving us warning. It says control missing segments pseudo bonds with pseudo panel. So this is just telling us that some residues are missing in this particular protein, and using this may cause some questions when we finally send our work out for review or for maybe molecular dynamics. So now to open the file that I downloaded, I'll just click on file, then click on open. It's just like any other file, any other software that we've been navigating so far. So now what we are going to do is we are going to look for the folder that I downloaded this particular protein that I just downloaded. So it's in my download folder. Sorry, it's in my download folder. And the name of the protein is, as you can see from here, it is CISDUK. CISDUK. So I'm going to select it from here, CISDUK. Then I click on open. You can see that this, from the PDB information that we viewed, it told us that we have about 5 or 6 chains for our protein. So, and we can easily see the structures of the protein are very similar. So we can easily see that they are incomplete. They are what I mean. They are homodimers. Okay, this is how many now? Maybe if it is 6, that is is it exam I should say now. But what I'm trying to say is this protein now, the parts it contains are similar. They are very similar. So we are just going to be using one of the parts for molecular docking. And we can see that all the parts, they contain their own co-crystallized ligand. As we can see from here, this is another one here. This is for this. This is for this. This is for this, and this is for this. So we are going. How do we select the one that we are going to use for molecular docking? We can use anyone. So to check, you can just click on select. When we click on select and we click on chain, you can see that we have 6 here. We can see that we have 6 chains. So we can see that we have 6 chains. So I will be using chain A for the purpose of this class. So click on select, then click on chain. When we click on chain, you can then select the chain that you want. I'm interested in chain A, and we can see that chain A will be selected. Chain A has been selected. When you click on that chain, the chain will be selected. So now that I've selected the chain that I want, I have to delete the other chains that I do not want. So how do I do that? I click on select once more. And when I click on select, I invert selected models. That is, I want to invert my selection. So when I click on invert my selection, you can see that it has unselect the one that I previously selected, and it is showing me every other proteins that are present. So to delete it now, I'm going to click on actions. When I click on actions, atom and bonds, and I come to delete. So we can see that I'm left with the one that I selected earlier, which is the chain A. So which is the chain A. So now I'm going towards the first step is now we know that there are no missing residues. So we have to remove the ligand that was co-crystallized with this particular protein. We have to remove the ligand that was co-crystallized with it. So we come to select, then we come to residue. Now this is showing us all the or everything that is part of our protein that are not amino acid residues. So in this protein, we are going to have water molecules and some other maybe other ligands as well. So when you click on residues and you click on all non-standard, this is HOH now, which is water, H2O. So it has selected all these tiny dots that we have right here. They are water molecules. So when you view them on PyMore, they will be more visible if you view them on PyMore. But we are using Chimera now, so we are going to be viewing them on Chimera. So if you to select one residue, then if you want to select water molecules alone, you click on water molecules, and if you want to select the other ligands that are present, let's say ANP now, or JBJ. So if you read the paper that comes with this particular protein, it will tell us more about the ligand, the particular ligand that was used to in defining the active site. So that ligand is this JBJ. The ligand is this JBJ. So and how do we save this JBJ? Because we are going to need it for our docking validation. So I've selected it by clicking on select residue and this JBJ. Then you are going to click on file and save PDB. When you click on file and save PDB, it's going to ask you for the folder you want to save it. So you navigate to the folder. So now I want to save it in download. I want to save it in download, then I can give it a name, maybe cognate ligand. Let me just say EGFR. So very quickly, I just click on save selected atoms only. I've selected a particular ligand, so I'm telling it to save the selected atoms only. Then I click on save. So that would have saved in my folder. So moving on very quickly, to get rid of every other thing except the protein, I will just click on residue, then click on all non-standard. So when I do that, I come to atom, bonds, and I delete every other thing. So we can see that what we have left is the protein and the protein alone. So now I'm going to go through the process that I said earlier, which is you add hydrogen, you do energy minimization and some other things. So to do that, there is a module on Chimera that helps you to do everything once so that you don't have to manually do it. Okay, I want to add hydrogen, I want to do this, I want to do that. So to locate that module, you are just going to click on tools. Then when we click on tools, we should note all these things that we have here, this red, blue, and yellow that we have here now. So we are going to see the difference after we do what is called dock prep. That is, we are preparing our protein for docking. So when you click on tools and you click on surface slash binding analysis, we also have it under structure editing, which is a minimized structure. You want to add hydrogen, you want to add charges. But all this process, they have been built into a module called the doc prep. So when you click on doc prep, it's going to do all these things that you are supposed to do one after the other. It's going to do them for us at a go. So we have it here for choosing molecule, the following: delete solvent, which is the water. If alternate locations keep only highest occupancy, okay, selenometionine to methionine. That is, if you have melters as part of your what's it called? As part of the protein, then it changes it to an amino. If it is selenometionine, you change it to methionine and some other things like that. Then incomplete side chains. If the side chains are incomplete, now these are not amino acid residues. So if we are talking about amino acid residues now, those are okay. Let's say maybe glycine is missing or histidine is missing. But for the side chains, it could be the H group in glycine. It can easily model that. It can easily fill that space. If it is the CH3 group that we have in alanine, it can easily fill that. So these are side chains that it can, and it has a library that contains the side chains for every amino acid residues that we have. But in case of in cases whereby it is the amino acid, the complete amino acid residues that it is missing. So we can't do it on a Chimera. Although if we have Modella, then we can do it on Chimera. But Modella is a commercial software, and we are focusing on free software. So we just do it using Swiss model. But for this class, we are going to okay. If we are focusing on the ones with incomplete side chains, if they are incomplete side chains, it easily replaces them and so on. It adds hydrogen. After adding hydrogen, it adds charges and it writes the Mo2 file, which I'm going to show us later on. So if we go back to us to this slide, we are going to see all these steps that I listed out as well, whereby we had hydrogen, we had charges, and so on. Then after that, we do energy minimization. So doc prep combines this, and when we click on OK, it does that for us. As you can see, rotamers, retrieving rotamers from Dunbar library and some other things like that. So when doc prep is done and when hydrogen has been added, we are going to see a change. Add hydrogens to it, then we click OK. When hydrogen is added, we are going to see a change in the structure of the protein now. Then it is asking if it should add charges. We click OK. So we can see now the structure, the hydrogens have been added. If we look at all these that we have here, they were not like this before, but now that hydrogens have been added, you can see that the structures have it changed a little bit. So to save this particular structure, we are going to save it as PDB. Then we can click on file, then save PDB. So we can click on file and save PDB. Then we navigate to the folder we want to save it. So we can name it EGFR doc prep. That is the EGF file that has been doc prepped. So we click on save. So now we are done with the doc prep part. So when I was talking about protein preparation earlier, I said okay. I said add hydrogens and charges, which we have done under doc prep. But now minimizing the energy of the protein. To minimize the energy of the protein, you have to come back to tools. And why are we minimizing the energy? So we are trying to obtain the lowest, we are trying to reduce the potential energy of this particular protein. So we are trying to bring it down to the lowest state, just like the ground state and the excited state. It is very much advisable to have it in the lowest possible state that you can have it, both your ligand and your protein, to have them in the lowest possible state for molecular docking, and that is why we are doing energy minimization. It minimizes the energy so that we can have them in the lowest state, and it helps the stability. When you take your molecule, your complex now after molecular docking, when you take them for dynamics, you want to check their stability. So if you dock without energy minimization, then it may result into an unstable complex. So to do that without talking too much, just come to tools. When we come to tools, we are going to click on this surface slash, okay, this structure editing. When we click on structure editing, then we click on minimize structure. We click on minimize structure. So now under for energy minimization, we have the steepest descent step and we have the conjugate gradient step. For the steepest descent step, what it does is that it's quicker than the conjugate gradient step. So what the steepest descent step does is it's just okay. Quickly do a search for the global minima now. It's kind of it's like a curve that I don't think we should be much interested in. But what it's trying to do is do the minimization, the energy minimization in a very much quicker step. But for the conjugate gradient, it's going to take much time to find the lowest possible state of this particular protein. So we are going to be doing both. But if you are interested, you can increase the number of a step that is the number of the. It's just like the time that will be spent in the searching for the lowest possible state. That is where this comes in. So we'll be leaving it in the default. Even at this setting, it will still take a little time. So we click on minimize, and when we focus on this part of the screen, I can right now my system is if you don't have at least a good system, it will take you a little time. My system is already hanging, but it's going to work at the end of the day. So it takes a little time for the minimization to complete. It does the steepest gradient descent first. Then after that, it goes for the conjugate gradient descent. So while this is loading, I will just open. I'll open PyRx. I'm sorry, my system is not that.
Hello, Victor.
Yes, sir.
Yeah, that's the part that Christopher was
Okay, okay. I'm sorry about that. Okay.
so you can um,
I will.
so thank you everybody. I will want us to I just need we will give room for just two questions because there is no time and the second speaker it needs to come up.
So please, if you have any question, I'll be happy to take them. And the ones I'm unable to take, we can just reach out to me. I will be happy to answer them on the group. So if there's any question, kindly unmute yourself to ask your question.
But I didn't know you please mute yourself. Alright. Can ask your question. Alright, so good afternoon.
Good afternoon. Yes, sir.
Yes, thank you very much for this.
Hello, can you hear me? Hello, can you hear me? Can you hear me? Okay. So my network is gone. Am I home? Sorry, please. I didn't get your question. Can you quickly repeat your question?
The question is, it said he is used to Discovery Studio in preparing protein.
Okay.
That people that are not used to camera, how do you think they can navigate themselves? What is the difference between the two, using camera and using Discovery Studio? And also, how can he navigate the camera?
Okay. First of all, navigating camera is very easy. I find it easier than Discovery Studio. I find Discovery Studio a bit tricky. It's a bit tricky for me, but camera is easy to navigate. Just load your molecule. No, what I believe is most of us we have navigated softwares that are harder than all these software that we most times used. We have interacted with web servers and websites that are harder than all this software. So you don't have to view Chimera as a complex software. it's something that you can easily navigate. You launch Chimera, you want to load a file, you click on file, you see the commands that will come out, okay, open, save, and so on. We all know what that means. Okay, you want to do energy minimization, you go to tools, then you go to surface slash structure editing. You are going to see minimize, you click on it, and so on like that. So it's very easy to navigate. But if you are having issue, you can feel free to reach out. So about Discovery Studio, so you are trying to achieve the same thing. So it's there are several ways to doing things. So it's not limited to one approach. So if you are used to Discovery Studio, it does not mean Discovery Studio is not doing a great job. But I'm trying to. I believe Chimera is easier to navigate, which is why I showed us how to use Chimera. So Chimera is a kind of easier to navigate. I believe Chimera is easier to navigate, but Discovery Studio and Chimera will still give you the same job. Will still give you the same result. So they will still give you the same results. You can stick to it, but if you are interested in exploring Chimera, I think you can just explore it to it. I don't think it's hard to navigate. I don't. Does that answer your question, sir? Hello, sir. Can you hear me?
Yes, yes,
Okay. Okay, sir.
yes. Marufat, please, you can ask your question. After Marufat, there is no more question, and we'll move to the next speaker. Yes.
Please, I have a question. I've raised my hand since.
Uh,
Can I?
kindly proceed. You can proceed. You can proceed with your questions.
I can only see Marufat raising her hand.
Yes, same thing here, sir.
Okay, okay.
But
Please proceed with your question.
maybe she's no longer here.
But somebody just said she's raising her hands.
She's the one.
Okay. Without much time, let us thank you, Victor, uh, for the presentation. I hope all of us enjoyed the presentation. However, the session is recorded, so at the end of the day, we will share the video for your user. Thank you very much. So, Mr. Chris Oloshoki, please, you have the floor.
Good afternoon, everyone. My name is Christopher Osayo, and I'm happy to be here today to give talk on the use of Python prescription for the molecular docking analysis. Before I start, I would just like to give a brief reiteration on what Mr. Victor said. For you to conduct molecular docking, there is need for you to identify a macromolecule which is either protein or nucleic acid, depending on
This thing, I believe this thing is easy to navigate software. Understanding what you are doing is
usually the small
that's what I believe.
peptide that is responsible
It was telling me 30
for
minutes.
carrying out the inhibition or activation of the protein of interest. Now, I believe we all understand the concept of central molecular dogma, which states from DNA to RNA and from RNA to protein. And then we all know that protein is the main byproduct or the end product that is responsible for every pathophysiology of disease in human. So the way protein is being expressed or regulated in the body will determine the kind of disease that will be generated or that will be influenced in the cellular function. So because of that, I'll just quickly define what an enzyme is. We all know that enzyme is a biochemical agent that speed up the rate of biochemical reaction by lowering the activation energy. And then these enzymes are responsible for like virtually all cellular process in the body. So for the concept of enzyme reaction in the body, there are two hypotheses that are recognizing that is the lock and key function and then the induced function. So because of that, we have been able to identify that before a macromolecule, that is a DNA or protein can exhibit a functional property in a cellular reaction. That means there must be an influence of a ligand, that is the small molecule. So now for molecular docking, this is a kind of in silico concept, a kind of computational concept that helps us predict how a ligand will interact with a receptor, that is a protein, in order to bring about a conformation that will elicit either an activation or inhibition process in the body. And then for molecular docking, it is based on two functions for it to occur or for it to be predicted. That is to be able to identify the binding position, which is the search function, and then to be able to predict binding affinity, which is based on the scoring of those functions. Now, there are different forms of molecular docking. We have the rigid docking, we have the semi-rigid docking, and the flexible docking. Now, based on the rigid docking, the hypothesis that is likened to the rigid docking is the lock and key function. And then the hypothesis that is based on the flexible docking is the induced fit function. And I believe many of us have seen publications where induced fit docking are being used. Are you with me?
We can hear you, sir. We can hear you, sir.
Yes, sir.
So now for induced fit, for rigid docking, now most of the old computational biology, that is docking software that are built, are based on the rigid docking. That is the conformation. I said earlier that the conformation of the receptor and ligand interaction is going to just be fixed. That is for rigid docking. That means there won't be changes. There won't
I want to download video on this thing. What's it called? Scrollinger.
ligand is going to be exhibiting different conformational posts for it to attach the binding site of a receptor, that is the protein or the DNA, depending on what you are using possibly. And then the flexible docking, our ligand at the same time our protein is going to be exhibiting a conformational change. So there are differences in these three forms of molecular docking. Now, because in reality, if a substrate or a ligand, for instance, want to interact with a receptor, ordinarily there is going to be conformational change in the receptor binding site. So in this kind of condition, it is likened to a flexible docking, which is commonly used for induced fit docking. So many of the docking tools we have today is only being carried out using semi-rigid docking for like the PyRx software that we are going to be using today. And that is because the protein of consideration that we'll be using in this software is going to just have a single conformation. That means there won't be changes in the conformation of the protein. That is the receptor that we are using. However, there will be change or rearrangement of the conformational structure, that is the spatial structure of the ligand that is going to be interacting with this receptor. That is the ligand that we are using. Now, there is something we call active site and allosteric site. Now, our protein of interest or our macromolecule of interest,
It's not using slide. We share his
eating it up.
You know him now, but you've read.
So we must be able to identify our active sites based on whatever we want to do. And I think Mr. Victor has been able to talk about that. So very quickly, I won't be going much into the preparation. Mr. Victor has talked about that. So please remember that for molecular docking, it is based on the principle of conformational change of receptor ligand interaction. If your ligand cannot conform, like fit very well to the receptor, that means your interaction, the binding affinity or the scoring function, the binding position of that ligand to that receptor will not be appropriate, and then the result that is going to be giving us will not be good. So now, because we are going to be conducting molecular docking, I've been able to prepare a protein already. And then the protein that we'll be using is the protein that is responsible for diabetic, that is SGLT2, that is sodium glucose co-transporter protein, which is responsible in the body for reabsorption of excess glucose in the kidney.
Hi,
I believe many of us know that till date, diabetes is one of the most studied disease globally, and then followed and also cancer. So I'm considering this because I know many of us here are working on diabetes as our research. So the protein I'll be considering today is the SGLT2, as I've said, and then the top chem ID is 7VS1. So I'll be sharing my screen now. So can you all see my screen?
We at Schrödinger have long believed that computer simulations can transform the way scientists design. Yes, it's coming up.
Okay. Now, as Mr. Victor have said, in selecting our macromolecule, we must be able to identify a macromolecule that is having a monodimers. That means it is having a monodimer, that is a single chain that might be in multiple, or it is just a single chain. So if it is having multiple chains that are different, that means you are going to be using everything together. But if it is having a single chain that is similar for all the chains, that is homohexamer or homopentamer, anyhow it is, you have to delete the other protein chains that are in the structure. So because of this class, I already prepared the protein, and then from the protein, there is need to identify the active site, because that is the what we are going to be using to predict the binding box at which our ligand of interest that we want to use to carry out a ligand receptor interaction will be docked. So if we don't know the binding site, or if it has not been experimentally determined, because when you download your protein from PDB database, most of the times you are going to see that they have crystallized ligand, that is the native ligand that has been used to study that kind of protein. So the conformation of that ligand in that kind of protein on that PDB has been predicted for you, just for you to be able to identify the amino acid residue, and then you write them out for you to be able to develop your binding grid, that is the box at which the ligands will be interacting with the protein. If that is not done, there are other softwares that can be used to do that. That is, you can use FT sites, you can use DocScope, and you can use several other web server, but that is not where we are going today. So because of this class, I said I've prepared the macromolecule, already identified the amino acid residue that is needed. That is where the active site of the co-crystallized protein is on the PDB database. So the first thing we are going to do, when we look at this interface, we are going to see that there are like three subdivision. That we have the navigator, we have the view, and we have the control panel. Now, one beautiful thing about PyRx is that although PyRx is not fully flexible and it is not fully rigid, because there are going to be conformational change. That is, the space interaction of your ligands will be changed to a fixed or a rigid macromolecule. That is semi-rigid or semi-flexible. So your protein is stable, but then your ligand is changing conformation. Now, one beautiful thing about PyRx, as I've said earlier, is that it is integrated with some functions like the autodoc VINA, because the program, the algorithm that is integrated into this PyRx is autodoc VINA, so you can easily conduct the molecular docking interaction of one protein with several ligands. You get me? So if you are picking a protein, Instead of you doing it one by one with just Autodoc, you can use PyRx to conduct a multiple ligand to a protein interaction with PyRx. And then another beautiful thing is that Mr. Victor said when you are downloading your ligands, your protein, the format at which you are going to be downloading them is in the SDF for the ligand, and then the PDB for the macromolecule, that is the protein. So if you look at this control panel, we have the Vina wizard, we have the Autodoc wizard, we have the Open Babel. Now, this Open Babel is a software that is being developed for you to convert the format, like a molecular format of a data, from one format to another. So, because we are going to be using ligand that are being downloaded in the SDF format, we are going to be using this Open Babel to convert them to the needed format, that is the PDBQT format, because it is always advisable to convert either your macromolecule or your ligand into the PDBQT format, because that is what this Python prescription recognizes for you to conduct molecular docking interaction between these two between the complex. So the first thing I will do is that I'm going to bring in my macromolecule, which is the SGLT2, that is responsible for the reabsorption of glucose in the kidney. And because it is responsible for the reabsorption of glucose in the kidney, there is need for us to develop or to find compounds or small molecules that is going to be able to inhibit the function of this compound. You know, when in a diabetic patient, are you listening, please?
Hello, Chris. I think
yes, sir.
Hidayat is raising up. Somebody is raising her hand.
Okay. Is there a question?
Please. Somebody raised their hand. Raised.
Okay. You can ask your question, please. Hello?
You can continue. I think the person that we can continue.
Okay. So you are still seeing my interface, right? The PyRx.
Yes.
So the first thing I'm going to load my molecule, that is, I'm going to input my macromolecule, which is the SGLT2. So I've been able to save it inside a folder in this place. Now you can see 4 different folder. That is the 7VS1, that is the one that was downloaded on PDB. The second one is the pre-process, that is the one that I've been able to remove out the ligand, remove the water molecule. Then the third one is the prepare, that is the one I've been able to add the necessary charges, add the hydrogen, and then minimize it as Mr. Victor have done. Then the third one is the native ligand, that is the fourth one rather, is the native ligand that was co-crystallized with the macromolecule on PDB. So I've been able to prepare that. So I'll be using this 7VS PK. So I click on it, I'm going to open it. Now you can see our macromolecule. Now this is our macromolecule for the 7VSI1, 7VSI. Now this format is in the PDB format. And like I said, before the PyRx can recognize this format for docking, we have to convert it into PDBQT, because that is the standard format that the Python prescription recognizes. So for us to do that, now this is molecule up here. I'm going to just left click on this 7VSI molecule. So we have display and we have Autodoc. Now in the Autodoc, you can see 2 things. It's telling me either I should make it macromolecule or a ligand. Because this is a protein, I have to make it a macromolecule. Then in making a macromolecule, it will be automatically converted into a PDBQT, because that is the recognized format for docking. So I'll click on make macromolecule. So this is it here. for me to identify if it has been converted to macromolecule. Initially, this is where I click on the molecules. I have to come to the autodoc. You see my ligand is empty, but my macromolecule down here is now showing me 7BSI prepared. So I want to know if it has been converted to PDBQT. I'm going to just right click on this. I hope you can all see that it has been converted to the PDBQT. Mind you, before I bring this macromolecule here, I already did all what Mr. Victor presented on. I already removed the non-amino acid residue. I removed the water, I added the hydrogen bond and the polar hydrogen bond, then I minimized it. So this is our macromolecule. Then the next thing to do is to input our ligands and the native ligand. Now the reason why I'm going to be inputting the native ligand that was retrieved from the structural format of this macromolecule is because it is going to be standing as a reference standard against our ligands of concentration. Now I said they have been able to experimentally develop that, and it is co-crystallized with this protein. But for our analysis, for our docking analysis, we have to use it because it is going to serve as a reference. So whatever our native ligand is, whatever the conformation that our native ligand is using to bind to this macromolecule is what is going to be considering for other ligands in this procedure. So I'm going to go to Open Babel, which is the web server or the software rather that is used to convert any format of the data into the required or the needed one. But because it is built with PyRx, it is only going to be able to help you convert whatever format you are using into PDBQT. So in this place, we are going to be adding the ligands one by one. So I'll be using one native ligand that is co-crystallized with the macromolecule. I will be using one standard drug that is canogliflozin that has been used that has been registered by FDA, and then I'll be using one natural product that is retrieved on a database. So the first thing I will do is now look at this interface of this control where my cursor is. I have the Vina, I have the Autodoc wizard, I have the Open Babel. So instead of wasting our time, I'll go to this place. It's telling me insert new item. So I want to insert my ligands. So I will insert the ligand here. Now the directory of my ligand is also in the same position where I input my, what are they calling it? The macromolecule that we are considering. So the first thing I'm going to include is the native ligand, which is in the PDB format. So I will include it. So I need to include this one by one. Now looking at this, you are going to see that the structural conformation of my native ligand, this is it here. You can see then the informations, the title, this is the native ligand, the formula, the weight, and the number of atoms. The next thing I'll add the other ligands. So this is the standard, that is canagliflozin, that has been approved by FDA for SGLT2 inhibition. So I will add it. This is it. This is how it looks like. So the next thing I will add one of my native ligand. Okay, let me add it here. I will add caralexin A1. This is it here. This is how it looks like. I will add caralexin A3. This is it. There is difference. I will add the last one, which is medicagonate. So this is medicagonate. So I have 1, 2, 3, 4, 5 ligands. And then from the ligand, one is the native ligand. Now, I've been able to use this native ligand to identify the amino acid residue for the active site. Like I said, if I don't have an active site, that means I'm going to be conducting a blind docking. That is, I don't know the site at which my macromolecule is either activated or inhibited. So that is why it is always advisable to download macromolecule with a native ligand so that you can be able to identify the amino acid residue that it is being crystallized with. Now, these are the ligands. The next thing I will do is that I'm going to minimize this ligands. Can you hear me, please?
Yes, we can hear you, but I think someone is disturbing from the background.
So we are going to minimize this like Mr. Victor have said, the essence of minimizing this ligand is for us to reduce it to
So,
Can you hear me, please?
yes, we can hear you, but one of the participants is.
The essence of us minimizing this native ligand minimum lowest potential energy. Said right from time that this docking process is a semi-rich because semi-fast. So the ligand is going to information that is going to the active side of the protein. So we have to minimize it to that is the lowest state before we can use it for this docking analysis. So I have to left click it. So instead of doing this minimization one by one, I'm going to click on minimize all. So when I minimize all, you are going to see that there will be changes in the structure, like the way the representation of this ligand are represented here in this view interface. It is going to change. So I've been able to minimize it now. You can see that it's reducing size. Before it was bigger than this. Are you with me? Now, because
Yes,
I've been able to minimize it, it has reduced it to the minimum lowest potential energy for it to be able to conduct the conformational change of the rearrangement of this molecule. So if it did not do that, it is going to interfere with the docking result. So we've been able to minimize it. Then the next thing for us to do is to convert it to the standard recognized format, that is the PDBQT. So I'm going to left click this. I'll click on convert all to orthodox ligand, that is PDBQT. I don't need to do it one by one. I can do everything at once. So I'm going to convert it all to PDBQT. So I'll click on this, I'll click it. So it is converting, it has converted it. Now, if you look at this place, earlier when I showed you, there were no ligands. So now, because I've converted it to PDBQT, you can see my native ligand. You can see these compounds, they have changed. The name have changed because one is canagliflozin, one is metacardinate, one is caralexin A1. But because I've been able to change it to PDBQT, it has changed to the standard PDB ID that these ligands are represented with. So you can always write that down on your when you are downloading it on PubChem so that you will not miss out the compound ID. So you write it out. So these are the informations about this ligand. This is the title. This is the formula of each of them. This is their weight. This is the number of atoms. Now, because I have converted this to PDBQT, the ligand, and then I've converted the macromolecule to PDBQT, the next thing for me to do is just to go straight to the docking analysis. Now, because PyRx is a very easy software, you don't need to start writing any script, you don't need to start putting anything. Everything has been integrated into the software. You just for you to click, convert it to the right format, click, and then it will do the analysis for you. So now, for us to conduct the docking, we have to come to this control panel, We are done with the open barbell. We come to the control panel. We are going to click on the VINA wizard. Now this is VINA wizard. Now from the VINA wizard, we have start here. So it is the program at which our PyRx is going to use to run. I believe when you are downloading this software, it has been integrated with it. So you are going to see it using your local program that has been integrated into the software. So it is on local. You get. So it is on local. The next thing for you to do is to click on the start. You click on the start. Now, when you look at this page, you are going to see that no ligand is selected and no macromolecule is selected. But because we need this ligand and the macromolecule for us to conduct this docking, we have to select everything out. So the thing you are going to do now is you are going to click on the macromolecule. When you look at this control panel, you see that it has changed to identify that we've been able to click the macromolecule. Now the next thing is you are going to click on control on your keyboard. Then you now click out each of these ligands. You click them. So when you click them, there are 5. Now it is telling us here that we have been able to select the 5 ligand, and then we have been able to identify the macromolecule that we want to use for the docking analysis. So now this is helping us. This control is used to us to select the multiple ligand for the VINA wizard, and then to select the macromolecule that we are interested in. So we have been able to select the 5 ligand, including the native ligand and the standard FDA recognized one, and the 3 ligand that we downloaded on PDB. Then the macromolecule. The next thing for us is to press on to the forward. Now you can see there is back here, there is forward. Then we click on the forward. Now a grid box is being generated for us. So this is where the work is. Now because earlier on I've been able to identify the active side of this protein. This is my protein. I've been able to identify it. So I'm not going to be conducting the blind docking. Now in blind docking, because I don't know the active sites, what I will just do is that I'm going to extend out this grid box for it to cover the entire macromolecule, because I don't know where it is having the binding site. So I have to let it cover it all so that it can bind to wherever there is a site for you to bind on it. But then, because there are other allosteric sites, as we all know, allosteric sites are similar to binding sites, but these are inactivated sites that are inactive. So it will not even hinder the function of the protein or the enzyme. So we need to identify the active sites in which this ligand, the native ligand in the body, are interacting with for this protein to carry out their function. Because if we do not identify this at the active sites, the essence of our docking will not be achieved. So we have to identify out these active sites. For the determining of the active site, I said they are web server, and another thing you can use to determine the active site is the Discovery Studio. Like one of the participants said, you can use Discovery Studio to identify the active site. That is using the downloaded macromolecule that is containing the native ligand. So I've been able to identify the active sites. So from what I have with me, the active site residue, that is the amino acid residue that is in the active site of this macromolecule, that is the SGLT2, are just 1, 2, 3, 4, 5, 6, 7, 8, 9, 11. Are just 11. So what I will do now is that for me to identify out the active sites, I will have to come back to this macromolecule. It was here before, that is in this navigation section, it is in the autodoc. So I will move it back to the macromolecule because that is where the amino acid residues of this entire macromolecule is. So you can see the amino acid chain is starting from ASP21 to the entire macromolecule is ending in 672. that is ALA672. Now, from my generated active site amino acid residue, I have TYR290. So I'm going to look out for TYR290. That is 290. So this is TYR290. So now I want to identify out this TYR290. I want to let it show me the atoms that are in this TYR290. Now, initially, you can see that inside this box, there is nothing being highlighted for the macromolecule. So I will go back to this TYR290. I'm going to left click this. So it is going to bring display for me an autodoc. You get. So I'm going to click on display, click on label, then I'll click on the atom. So when I click on the atom, you can see that has been highlighted. So if I go to this view interface, the 3D scene of my macromolecule, you are going to see that the atom will be highlighted. You can see it. The atom for the TRY290, these are the atom, and that is where it was predicted using my discovery studio that the active site of this macromolecule is. So the next thing is, in my active site, I have GLU99. So I will have to come here again, look for GLU99. So this is GLU99. You can see as I click on it, it brought out the atoms. So I have to identify it out. So I have to click on it so that it will show me that I've been able, yes, I've done that. So the next residue that was predicted was PHE98. I'm sorry I'm not showing you guys that. So this is it, PHE98. As you see, as I identify it, as I click on it, it is being identified in the view section. So I have to click this one too out. Click on the atom. The next one, let me just do two more. I have ASN75, and I have GLN457, 457. Now this GLN457. You can see it has been identified out. So now, because I don't want to carry out blind docking, I'm now going to reduce this my grid box to this area of my atom that has been identified out. So I'm going to reduce it. I will just right click it and reduce it like this. So because this is a 3D structure, I have to turn it around for it to just identify out those section I pin out alone. So it will not bind to any other binding site in this macromolecule. Now let me look it. Okay. So this is it. I reduce it. Now if you look at this grid box now, it is containing just the area in which I've been able to identify the amino acid residue atom in the macromolecule, because I've been able to use Discovery Studio to predict that. So I'm not going to be binding or conducting the molecular docking to just any binding site in my macromolecule. So this is just the place of which my ligands, all the 5 ligands, will be interacting with this macromolecule. So every other place is now of my concern, because that is not where it it was predicted that the binding sites, active sites rather, not binding. So this is the binding pocket. But then the site at which these atoms are identified, like this A, like this CD1, like this CB, like this H, like this N, those are the sites. So this is the pocket, each of these atoms is the site. Then this is a grid box. This box is called grid box. So it is directing, or it is helping my ligand to know where they are going to interact or to form conformation with just this macromolecule. So it will not interact with any other region on this macromolecule. So the next thing now that I've been able to do this, I've been able to identify that out. I can reduce this, increase this. Now you need to write, maybe for publication purpose, you can write out this space information. You have the center and you have the dimension in abstract. So you can write it down, you can include it in your publication for the purpose of other people that might want to use your macromolecule in docking study. So now we have the exhaustiveness. Ordinarily, the exhaustiveness is always in it, so you don't need to worry about that. So you leave that there. You write this one out, then the next thing you just do is to press on forward. So your docking procedure starts.
So thank you, Mr. Christina. Let us use this time for questions and answers before the docking. Okay. Mr. Adedoyin, please ask the question.
All right. Thank you very much, Mr. Christopher. Okay, my question is, I've done something similar to this. I mean, using this particular software and to do to molecular docking. But I realized that I had issues with the docking process. Like after, like the way it's running like this now, it's just showing error. Like I don't know. So I was trying to figure out where the problem is. Maybe it was people I didn't prepare to put in very well, or like I didn't know where exactly the issue. So in case one experience something like this, how is it possible to troubleshoot where the problem is? Something like that.
Okay, like you have said, if you did not carry out the necessary procedure, that is, you include your charge, you minimize the protein and the ligands, and then you convert it to PDBQT, you might not get this the whole thing. And another thing is that when you are downloading your protein, Mr. Victor have said most of the thing I am supposed to see. Because if you are having let's say non-amino acid residues that are in the macromolecule, and then in the process of you pre-processing it, that is in the stage at which you pre-process it and you prepare it using either Discovery Studio or Chimera, and you meet one of these parameters that is essential for PyRx to identify that this is a format it can read, the process will not be able to be conducted. So if you go and look at your what are they called it, your molecule again, it is either that your macromolecule that you are using for the docking is the one having issues. Like he said, there are some macromolecule that you download online that there are some amino acid residue are missing, and those residue are essential for the interaction, that is the binding interaction between the macromolecule and the ligand. So if you are downloading this kind of macromolecule for your docking, you are actually going to have problem. So that is why it is always advisable to look into literature to identify proteins that has been commonly or regularly used for any kind of analysis or project you want to do. So you don't just come up with one protein from one place, except otherwise you have experts that is going to help you maybe develop the protein that has not been done before using homology modeling, as he has said. So I think that's where your issue is. You have to go back, look at the information about the protein that you are downloading, check if they are homodimers or heterodimers. Identify the binding site in that amino acid. And then if it is not having a binding site, that is because it is going to be crystallized with a ligand. If it is not having that, there are sites at which you can identify the binding site, like the FTP site, like the Oxford site. So, and then there is one other site, plus scope site. So you can use those sites, those web servers to predict your amino acid, so you can get rid of that error. So that is it, sir. I hope I have been able to answer your question.
Yes, sir. Thank you very much.
Okay.
There's a question in chat box.
Okay. Okay. How are we choosing the amino acid in the macromolecule for the binding with the native ligand? Yes, as I've said, you have to identify macromolecules from literature that have been done very well. And then when you identify macromolecule, it is not that most of the time they will tell you that this is the binding site. That is what I said. Whenever you are conducting your docking, you can write, look at this place my cursor is. You can write out this center information, this dimension information for any other people that might want to use the protein or the macromolecule that you are using in your analysis. So it will be easier for them to use these coordinates for them to for their docking interaction. So if you don't have that, if you are not able to identify that in the publication, you can always use web server to predict the binding site. That is the active binding site of your macromolecule. Like I said, it's FTP site, there is core site, there is plus site. So there are various sites online that you can use to predict the active site online to locate it. So if you have been able to identify the binding site, in that kind of situation, that means there is going to be a native ligand crystallized with that macromolecule. So it is that native ligand, because it has been experimentally validated that this is where it will interact with the macromolecule. It is that native ligand that is going to predict the amino acid residues that the ligand is interacting with on that protein. So that is where you are going to use to identify out the amino acid in your macromolecule for you to draw your grid box. If you are not able to draw the grid box, that means you are going to be conducting a blind docking. And then your ligand can interact with any site on the macromolecule. You know, there are binding sites, active sites, there is allosteric sites. And these sites are, some are semi-competitive, some are competitive. But then if it is not binding with the active site, that is the site at which it will inhibit or activate the functionality of the macromolecule, that means you are still not doing anything. That is why it is essential that when you are conducting docking, you have to know your binding active site. I hope you get it, sir. So you can get this when you are downloading your macromolecule on PDB. So if you are downloading any macromolecule, be sure that your macromolecule is having ligand. And then there are instances where you download the macromolecule, you have more than one ligand in it. Now, sometimes you are going to see metal, sometimes you are going to see compound itself. Now, identify the ligand in that kind of molecule that is having the most interaction with that molecule. If you are having more than one ligand in the macromolecule, you must be able to identify the one that is essential for you to receive the functionality. Because if you are not using the correct ligand, you can also have issue in that kind of situation. So it is better to look for ligands that are recognized. And another way for you to be able to notice is that if you see multiple ligands in the macromolecule, go to literature, search on those ligands' name. You are going to see if they are the one that are being used regularly, or it is just a new ligand that is crystallized with it. So you don't need to bother about that. You can only search the name online. You will see if they have been using it, or it is a new ligand. So that is it.
I think Marufat wants to ask a question. She's raising her hand.
All right, your question.
Thank you for the presentation. Please, I'd like to take the question from where you stopped about the molecule, the ligand, the active site of the thing. So I think, just now you try to summarize how it's been done. So after getting the ligand that was, reacting with the protein, I think for us to do, we have to know the ligand itself, right? The ligand will not be present. Right, so,
Yes, you have to.
I have a question. Hello?
Okay, I'm listening.
Okay. And this issue of homodimer, all these things, during the first presentation, and you also repeat the same thing, they are trying to say there are some, ligand, I'm talking about the other one. There are some ligand that we have like 4 to 5 chains, let's say 6 chains, and we have to look at this in like just one of them.
You mean protein?
Yes, no, not the protein. I mean the ligand itself.
Okay.
The
Conformants, you mean?
The phyto compound or the artificial one.
Okay.
Yeah, that is what I'm talking about. So that whereby they now have like 1 to up to 5 chains. Now so we have to look at it. How do we see just by mere looking for us to know if they are this
Okay. Now the homo we are talking about, is not for the.
How will we confirm that we are similar again? We should go for just one of them.
The homo Mr. Victor talked about is on the protein, the macromolecule, not the ligands. Because, like, virtually all proteins, let me not say all, some proteins have amino acid residue and then the side chain. Now, this side chain are not all sometimes responsible for the activity or the functionality of the protein. Now, if a protein is homodimer or homodimer, that means it is having 2 side chains that are similar but they are double. So in that kind of situation, when you view it, you are going to see that the structure of those kind of macromolecule is the same. Where the ligand is binding to those 2 residue, it is the same. So it is essential for you. You can delete one, you see chain A or chain B. Everything, chain A and the chain B, they are the same. So if they are the same like that, you don't need to use the 2, you have to delete one. But then in a situation where they are heterodimers, that means they are more than 1. That means they are 3, and the 2, 3, but they are different chains. So you have to just identify where the active site is on those chains, but you don't need to delete the other chains. I hope you are getting me. Are you with me, ma'am? Hello?
Yeah, I'm struggling to hear your voice.
What I'm saying is that on PDB, you can have a macromolecule that have more than 1 chain. You will see chain A, chain B, chain C, chain D. In some, you see some that will have chain A and chain B. Now, if they are homodimers, that means it is having 2 side chain, but that 2 side chain is similar. They are the same. You get it? Now, in that kind of situation, you will just need to delete one because the native ligand that has been co-crystallizing that is the same thing. The side chain is the same. You have to delete one. But in a situation where they are heterodimers, you don't need to delete all. You just have to identify which part of the side chain or which of the side chain is it A, B, or C that is having the active site. That is where you will see the native ligand. So you cannot see the native ligand on multiple places in an heterodimer, but you can see it on homodimer in multiple places. The same native ligand, you can see it in multiple places in homodimer. So if you are having a heterodimer, just identify out the active sites. Then you are going to clear out the water molecule, clear out the non-amino acid residue. Then you can save it and use it for your analysis. I hope you get it clear.
I agree with the integral part, but if they are home and they have many cheese, my question is just that: is there anything for farm? As in, is there any software for us, or we just confirm by mere looking, by mere seeing the all the change look the same?
Yes.
Look at it, and that's
When you be it on PDP, you are going to see the information about it. If it is homodimer, you are going to see that side chain A and chain B. You can easily see if they are the same on PDP. But if you cannot identify,
virtually, I mean, with our eyes.
yes, you see it. You can, like I said, look at this PyRx now. Let's say, for instance, I didn't prepare this amino acid macromolecule rather. So let's say this is TBS1, and we are going to see chain A. It will start from ASP21 down to ALAC7. Then you are not going to see chain B also beneath it. It will start from this 21 to 627. That is how it will look like. So there is no need for you to use those side chain that are similar. So we have to delete one out from it.
So, Mr. Chris, the result is up. So, is there anything you want to add? Because we want the next speaker to take over.
Now, like I said, PyRx is a semi-rigid, semi-flexible analysis. Now, our amino acid, our macromolecule is rigid, but the ligand is going to be like conformation of the ligand is going to be changed for it to have a post that is able to interact very well inside of this macromolecule. Now, one of the things we used to consider for us to pick the best binding affinity is the RMSD, that is the root mean square deviation. Now, from literature, it is said that the lower the RMSD, that is from 2 downward, the better the docking score. And that is why most of the time you are going to see that the docking score that is peaked or that is mostly peaked in the binding affinity that is mostly peaked in docking analysis is the one that is having the RMSD 0. That is for the upper bound and the lower bound. That is root mean square deviation. Now, in the binding affinity for the native ligand, you can see that the affinity is minus 9.6. Now, this is a conformer for the native ligand. This is now looking at this view. When I click on the other information on the native ligand, you can see that it has changed. The conformation has changed. The conformation is changing, but then the macromolecule is rigid. It is not changing. Ordinarily, in a cellular atmosphere, it's the conformation of this macromolecule is supposed to change, but then it is not changing. That is why PyRx is a semi-rigid, semi-flexible procedure. So we can see the conformation of the native ligand. Now, this is one of our, what are they calling it, compound. Now, the native ligand is having minus 9.6 binding affinity. Our compound is having minus 10.2. That is to tell us that the native ligand that is the reference, the binding affinity to this macromolecule is lower to our compound of reference, that is minus 10.2. So in this kind of scenario, we can say that this compound is better than our native ligand because it's having minus 10.2. That is for the RMSD at 0. So as you look it down, you are going to see that the binding affinity reduced based on the RMSD formation. So the conformation is changing also. The conformation is changing. The conformation is changing. And then the other one, this is another native ligand. We have minus 9.1, which is closer to the native ligand on the RMSD 0. Then the last one, we have this, which is 10.2. You can see this information is not what we are expecting. So this is 10.2. So that is how we are going to pick out our binding affinity that we need. Now the last thing for you to do is you need to save this table. So you can always go back to it. So for you to save it, you are going to come here. This is where you first add the ligand, see the delete current. This is set up reference for table, and then save as command separated value. So you click on this save as command separated value. You navigate down to the folder you want to save it. Then you can now name it docking results. Now this has been saved. This information on this table has been saved. Now the next thing you need to do is that for you to conduct your 2D 3D molecular interaction, for you to see how your ligand is interacting with this macromolecule, you have to export the information on this view interface. And for us to do that, we have to come to this file. We click on import. Sorry. We click on export rather. Come to the file. Click on export, and we want to retrieve the conformational information that this docking has been able to generate for us. So you navigate to where we want to save it, go to the directory, come to workshop, you can save it. This is PyRx Orthodox Star, you save it. So you click on finish. So the information will be exported to your directory. So it has been successfully exported. So you click on OK. So for your 2D and 3D molecular interactions, we can carry that out with that. So the next presenter will be giving us more insight on that. So thank you.
Thank you very much, Mr. Chris. We really appreciate the effort you put in explaining the techniques of molecular docking using Python Precision 0.8. Victor.
Yes, sir. Yes, sir.
Yeah, I think you have leak plot with you, right?
Yes, I do.
You do, okay. Real one is on transit. So unfortunately, he cannot get the car on time. So you please be taking that aspect to the 2D interaction using leak plot.
Okay. Okay, sir.
Thank you very much.
Yes, sir.
So you own the floor.
All right. So thank you, Mr. Chris, for the wonderful presentation. So just to move on very quickly, we'll be proceeding with the 3D visualization. So what that is simply trying to do is, now we have, after docking, we are going to get a number of poses, like number of, let me just show us a docking result. I will share my screen so that we can have a better understanding of what I'm trying to say. I'm trying to share my screen. Is my screen visible? Can we see my screen? Okay.
Yes, it's loading.
Okay, sir. So I will just navigate to a folder that contains some of the results I've obtained in the past so that we can easily have an understanding of how things work. So now, this is like a representation of what we are going to have after docking. So this is like this docking result that Mr. Chris was telling us to save as CSV. When we open this result, it's going to show us the scores of our poses. But what we are interested in right now is not that. What we are interested in, if we now this one, if we are launching PyRx for the first time, it's going to save the information of our docking. It's going to save it in the folder. You can just try and get where that folder is located. So and how to do that, just if you go to PyRx, then go to edit preferences. I think let me just launch PyRx and quickly show us that if you are trying to know where your work folder is on PyRx. Sorry, I'm trying to launch PyRx, but it's a folder that will contain what we have right here on my screen. So that folder will contain it, and I'm trying to show us how we can navigate to that folder. So when my PyRx comes up, I'm going to show us that. But moving on, we are going to have this etc, then we are going to have this ligand. Now this ligand, it contains all the ligand that we imported initially when we wanted to start the docking. So these are the ligands, and they are saved in their PubChem ID and the value of their minimized energy. So we are not concerned with this for visualization. We are concerned with the macromolecules folder. So when you open the macromolecules folder, we are going to see all these. So now, this is the macromolecule that we used for docking, which is our protein. When we, as like Mr. Chris demonstrated to us when he was drawing the docking, he said we should click on autodock, then make macromolecule. So when we make macromolecule, it will create a folder. So and that folder is going to contain the macromolecule that we choose. So after that, all the ligands that we imported, after the docking is done, these are the poses of all the ligand. This is the first ligand for this particular result that we are working with now. This is the result of the first ligand, and we are going to have, is it 10 or 8 poses now? We are going to have about 10 results for this particular ligand. No, Mr. Chris was talking about RMSD values. So the when after docking, it's going to generate several poses. So the one with the highest score is the one that we usually select. So, and how do we do that? And that is where we are going to navigate back to our PyRx. So I'm trying, I don't know why my PyRx is not coming up, but kindly give me a minute, I'll fix that soon. Okay, now it's up. It's still loading.
So,
I'm coming, I've opened it multiple times, which is why the error is coming up. PyRx. So now just another bit of information. So Mr. Chris was talking about like the XYZ coordinates that contains our ligand earlier. So during the course of the docking, the algorithm will save that coordinate itself. So the algorithm will save that coordinate. So when we go to our work directory, which is the folder that contains our docking, the Pirates that we created for our docking, when we go to the macromolecules folder, we are going to see something like con conf, like conf. So it is in text format. It will be in text format. It will contain all those coordinates, like the XYZ coordinate, the and some other information. So let me just open that so that we can have a look at it. Okay, so now we can see that we have it here. We have the exhaustiveness, which was 8, the center X, the center Y, the center Z, size X, Y, and Z. So we can put it easily, come to this place when we want to write our manuscript and put it in the manuscript. So moving on, I have my Pyrex up. So now to get where your work directory is, just come to edit. When you come to edit, click on preferences. When you click on edit, it will show preferences. So you click on preferences, and when you click on preferences, it's going to show you where your workspace is. If we can see my screen right now, we can see that we have workspace here. So and it will show you now. This is the when you just install Pyrex, this will be your default workspace. This will be your default workspace. So if you are working with a particular, you want to dock, every information about that particular job that you did will be saved in this workspace as a folder. But you can change it. Okay, maybe I'm working with like this is the way I do my. Okay, I want to work with EGFR now. So I'll just create a file. Okay, I'll create a folder docking, and I'll okay EGFR docking. Then I can you now copy the directory. So I'll be showing us that right now. So if you want to change your workspace, I just want to quickly show us that you just click on browse. When you click on browse, now you want every of your information to be saved to a folder of your choice. You just navigate down to the folder. You navigate down to the folder and you click on OK. So I won't be going through that. You can make a new folder. You can just select a folder that you already have, maybe in your music, and you create the folder. Okay, docking, and you click OK. So you can see that it has automatically changed the whole thing. So after docking, that's where you are going to have something like this. That's where you are going to have something like this. You are going to have the etc. You are going to have the ligands. You are going to have the macromolecules. So now, if you click on the macromolecules, click on this again. You are going to have every information that you need for your visualization. We have the poses that were generated after the docking. So now I had 10 compounds here for this particular docking. So for each of the compound, you are going to have about 10 poses for each of the compound, and you are going to select the number one, which is always the best. So quickly showing us that now, I'm going to open this folder on PyRx, click on File, Load Molecule, then you navigate down to the folder which is your workspace. No, now I have copied the path, so I will just paste it and click Open. So this is a compound. When I open this compound now, when you click on Open, you can see that these are the models that it generated in the course of the docking. We can see that we have 9 poses here. So we are only interested in one for visualization. So, and the first model is usually the best. So you are going to select the first. So you are going to uncheck the other ones. You uncheck the other ones. So when you uncheck the other ones, this is the only one that we have left. You right click. When you right click, you save as PDB. You save as PDB. So navigate to the folder you want to save it. So I'll be saving mine in my pictures, in this particular folder. Then you are going to name it. Okay, you can just say maybe you are in the list of your compound. Maybe stigma steroid is the best. You can just say, okay, stigma steroid.pdb. Please take note of that. It has to be in .pdb. So let me say this is a quercetin now. I'll just say quercetin.pdb. Quercetin.pdb, and you save. So if you go to that folder where you just saved it, you can see that you have it. Now, Pymo is my default software for opening PDB files. That is why you have this logo. If Discovery Studio is your default software for opening PDB files, you are going to have Discovery Studio logo here. So moving on, you are going to if you want to visualize four different compounds, you are going to repeat this process that I just completed. You are going to repeat it for the 4 compounds. I hope we all get that. So after that, then you can then do away with all this. You can remove from scene and so on. But I won't be able to do this because of our time. The next thing I'll be doing is I will load the macromolecule that we used for molecular docking. The macromolecule that we dock prep and did energy minimization for is what we are going to import again. So you click on load molecule. Now, how do you get that macromolecule? That macromolecule is this. It is also be in that macromolecule folder. Let me show us once more. We can see this here. This was the name of my home macromolecule when I was doing this docking. So this macromolecule folder will contain both the configuration file, and the macromolecule you used with all the poses that were generated after the molecular docking. So you are going to open this macromolecule now in PyRx. So to do that, you just come to PyRx, then you click on your macromolecule, which was the one that Mr. Chris emphasized that you click on auto doc, you make macromolecule. When you make it a macromolecule, it will change it to a PDBQT file, which is what we have here as .pdbqt. So you click on open. When you open it, it's going to display. This is it right here. This is it right here. Then you save it as PDB as well. Now remember, it was formerly in PDBQT. For visualization, you have to convert it back to PDB. So you save as PDB. So you can navigate to the folder where you saved the ligand that you want to visualize with it. So let's say the protein was okay. Let me just say EGFR. So now .pdb again. So now you are done with what you need PyRx for. So you can close PyRx. So moving on, you navigate to the folder where it was saved. Now these are the two molecules that we need for our visualization. So open it in PyMore. Open it in PyMore. Now PyMore is my own default software for opening PDB. That is why it opened in PyMore directly. It will open in PyMore directly very soon. But if your own default software for opening PDB files is Discovery Studio, you have to open it with PyMore instead. You right click, then you click on open with PyMore. I'm sorry, my system is always a bit slow due to the loads on it. It will open very soon. I'm sorry, this is taking longer than I expected, but okay, now it's up. Now this is the molecule that we use for the docking. You remember we remove during the dock prep process, you remove the cognate ligand, you do energy minimization, add hydrogen, and all sorts of things like that. Then you convert, you repeat, you go over the process that I just completed. So after that, you open in PyMore. After opening in PyMore, then you load the the ligand that you saved separately. You load it. You click on open. You want to load it separately. You are going to load it together with this protein. You are going to load it together with this protein. So this is the ligand. You load it together with this protein. You can see that now what this will give you, it will be when you after your molecular docking, your compound must have bind to your protein in a particular active site and in a particular conformation. So that conformation will be retained as long as you follow the process that I took earlier. That conformation will be retained. So it's that now you have your complex, which is your protein ligand complex. Now it is EGFR quercetin complex. So now we have our complex. This is the shape that the conformation that are compound that it's bind to that the conformation is formed in the active site of this particular protein that we are working with, and that is what we have here retained. So now you are going to save it as a complex, and to do that you are going to click on file, then you click on export molecule. When you click on export molecule, you are going to see the selection. You are going to see something like enabled. So you click the drop down menu. When you click the drop down menu, you click the whole. No, we are trying to save them as a complex now. So when you save them as when you click the whole, then you click on save. You click on save, then you name it. Okay, EGFR. Let me just use it's not a good practice to use a capital letter. I just learned that recently when naming your complex. Let me just use shorter names. Shorter names are very important. I'm working with quercetin. I will just give it a short name, QT. And I'm working with EGFR. I will just give it EG. So EG QT complex dot pdb. You are also going to save as pdb. Just to go over things again, you save the PDBQT of that the macromolecule that you made as PDBQT. You first open it in PyMore, save as PDB. The best post of your compound, you open it in PyMore, save it as PDB. Then you complex them again and save as PDB. That is, you open them together and save as PDB. And that is what we have here. So it would have saved in my folder. Now we have it here, EGQT complex. So you open again in PyMore. You open again in PyMore. So now, what are the things that we are going to be needing when it finally opens? The first thing is you have to get, okay, what are the amino acid residues that were used in identifying the binding site? You are going to need it for the visualization purpose. You are going to need the amino acid that we're using in the molecular docking. So we can see we already have our ligand here. We already have, this is our ligand and this is our protein, and this is the complex that they formed on docking. So now to proceed now with the visualization, come to click on actions, then you click on center. That is the first thing. So when you click on center, you'll be able to action and zoom. So when you click on that, I'm coming late. Okay, I'll come back to that. But when you load it, just come to display. The first thing we have to do is click on this, uncheck some of the things that we have here. This is just to ensure that we have a fine representation of our complex. Uncheck the show valencies. I was trying to zoom the complex earlier so that we can see what all these things are doing. But okay. Now let me just show us again. Now look at this here. All these like how do I describe it? Like it's just like a structure is it contains some other ring inside. So that is one we want to do away with. So you can click on display and uncheck the show valencies. You can see that it has disappeared. After that, you uncheck this as well, which is the depth Q. You uncheck it as well. So after that, the next thing is to select our ligand. This is our ligand. To select it, you are just going to click on it. But I'm sorry, my system is a bit problematic. Okay, now we have it. You just click on the ligand once. When you click on the ligand once, then you rename the ligand. So when you click on it, you would have seen something like selected here. So which is SELE. Click on actions, then rename selection. That is our ligand. So after that, we can just uncheck. So now the next thing is to select the amino acid that were involved that we used in the molecular docking. That is the amino acid of the active site. So how do we do that? First of all, we have to display the sequences of our protein. You click on display, then you click on sequence. So you can see that we have it here. These are the sequences of the protein that I worked with. So you now select the amino acid that were in the active site. So now, okay, let's say this was involved in the active site. When you click on it, it will select it. I'm trying to get the amino acid of this, but I'm not with the book in which I wrote them. I'm very sorry for that, but we are just going to work with some other amino acid. But just ensure you select the amino acid in the active site of the protein. So let's say this is in the active site of the protein, and you use this in your docking. You just select it by clicking on it. A, it will select it. Then you also select by clicking on T. So in this manner, you are going to select all the amino acids that were involved in the that are in the active site of the protein. So you continue doing that. You are going to have everything selected. So in this manner, you are going to select everything. And when you after selecting everything, you just come back here. There should be something like selected. I'm coming. I'm sorry, there is a bit of issue. All these things that are currently showing, they are not supposed to be showing. Like all these PK1. So you have to get rid of them if you have issues like this. And all these PK more, PK recipe. Just ensure you get rid of them. So let me select our amino acid again. You can see that the selected ones have come, they are already coming out, and we have it here. This that we have here. So let me select other ones. The select every amino acid in the active site. So moving on after that, you are going to rename the selection as well. They are the active site amino acid residue. Active site AA. That's active site amino acid residues. So now after that, what you are going to do now is give your ligand a color. Give your ligand a color. So you come to this that we have here. This A means action. This S means show. H means hide. L means label. And C means color. So now we are trying to give the ligands alone. We are trying to give them a color. So you are just going to click on the ligand. That should have appeared, but my system is a bit slow. So you click on C, which is color. Then you click on by element. So you can now select the color that you want. I will be going for the first one. System. Sorry, there was an issue earlier. The ligand that I saved didn't save it. Something else saved as the ligand. So I'll be deleting this and re-click on the ligand. If you have a good system, you won't be facing all this issue. My system is slow. I'll be coming back to this. I'm currently experiencing some issue with this particular selection, but we shouldn't worry about it. We are going to get the process involved. So the next thing I'm going to do is click on this complex, then click on a preset. Click on preset. After clicking on preset, I'll click on ligand site. When I click on ligand site, I'm going to load the ligand site as cartoon. We are going to see the effect of that now. So the reason why this is not really showing as a cartoon is because the active sites that I selected are different from the one involved in the interaction. But all you need to know is you click on the complex. When you click on the complex, click on A, then you come to preset, then you click on ligand site, then you click on cartoon. So we can see it. It will bring out all the active site amino acid residues. So that is what we have here. All these that we have here, they are the active site amino acid residue. You can see an hydrogen bond here. Let me zoom in a bit. This is an hydrogen bond, another hydrogen bond. So to go over the steps again, this we are not done yet. But the first thing is after loading your complex, you click on action. When you click on your ligand, you rename your ligand. You click on the ligand, you rename the ligand as your ligand. Then you select the amino acid residues that interacted with the ligand. That is the amino acid residues at the active site. You rename them. After you rename them, then you click on the complex. You click on action. When you click on action, you click on preset. When you click on preset, you click on ligand site. When you click on ligand site, you make it a cartoon. After making it a cartoon, then you look for the best pose. That is the position that will show the interaction clearly. The position that will show the interaction of your protein and ligand clearly. That will make all the amino acid visible. You click on it and maintain that position. So we'll be going with this. We can see the amino acid at the active site are showing. We can see that they are showing. So we are going to reduce the transparency. That is, we want to make the only the amino acid at the active site obvious. We don't want to make any other amino acid residue obvious. So we click on settings, then we click on transparency, then we click on cartoon. We reduce by 80%. If we are following, we can see that this has every other amino acid residues apart from the ones at the active site have been sent to the background. So we can see them here. They have been sent to the background. So the next thing we are going to do is change the color of our ligand. Let's try that again. Invalid selection. My ligand is having some issue, but we'll be moving on with it like that. So when you click on ligand, just click on show. So we are going to be showing our ligand. We are going to be showing it as sticks. Sticks. I'm trying to select the ligand again. We'll just proceed. I'm going to get back to us on why this particular issue is reoccurring. But the next thing we are going to do is change the color of the color of our protein. We are going to change the color of our protein. So we are going to first of all go to settings, then we are going to go to cartoon. Now, just to illustrate, clarify something, the every amino acid, the structure of the protein is represented as the cartoon. So when we talk about cartoon, I'm talking about um, all these alpha and the beta helix that we have here. They are the one we are referring to as cartoon. So click on settings, then cartoon. When we click on cartoon, we are going to click on fancy helices. We are trying to make them fancy, just to like making them more beautiful. Let me just illustrate that a bit more. Settings, cartoon. Now, when it is not a fancy helix, we can see this that we have here. Let's look at this shape that we currently have here. But when we make it a fancy helix, cartoon, then fancy helix, the orientation will change a bit. Then when we after doing that, then we click on settings again, we click on cartoon, then we click on highlight color. What that is trying to, we are trying to make the colors of the internal part of the chains, we are trying to make them uniform. So we can see that the colors are now uniform, they are now more of a black, they are now more of black. Then we click on settings, we click on cartoon again, we click on cartoon, then we click on discrete colors. Let me show that again. Settings, cartoon, discrete colors. So now what the discrete color is trying to do is we are going to see that when we give our cartoon a uniform color. So I'm just going to proceed, setting cartoon, then discrete color. So now I'm going to come back to the complex, then I'm going to give the complex a different color. Let's say orange now. So I've changed the color of the whole complex to orange. Then let me try this again now. Now, the first after changing the color of the whole complex to orange, then I was able to change the color of the ligand to the to by I was able to color the ligand by element. I was able to color the ligand by element. Now, how do we make the amino acid at the active site different from every other amino acid? So to do that, you are just going to come to the color again under the active site amino acid residues that we currently have here. Then you click on color, which is C. When you click on C, you are going to color by element. Let's click on color by element. Let me just give it a different color. Now, the reason why this is not showing is because the amino acid that I selected are not the ones in the active site. Like I said earlier, I don't have the ones that I used for the interaction or for the docking. I don't have them here with me. But if we look at what we have on the screen right here, we can see that some amino acids are currently colored red. So if they were the amino acid in the active site, the color would have changed completely. The color would have changed completely. So when we, after selecting the amino acid and giving them a new color, then we can now label them. And to label them, we are just going to click on display. No, we are going to click on settings, and we are going to click on label. When we click on label, sorry. Okay, I'll move to the 2D interaction very soon. I'm trying to look for leak plots on my system, but I'm almost done with this. just click on wizard. So the main cause of the error is because the amino acid at the active site are not the ones that I selected. We would have seen the names of the amino acid. They would have come out on our screen if they were the ones at the active site. They would have come out on our screen. When we click on wizard and click on label, the sequences would have been displayed. The sequences would have been displayed, but because they are not the ones selected, that is why we don't have them out here. But on a normal condition, they would have, if this amino acid was at the active site, this amino acid would have been given a name, and these other ones would have been given a name, and so on and so forth. So after giving the residues their name, we are just going to make the background white. So to make the background white, we come to display, we come to background. When we come to background, we click on white. So it's going to make things white. Then we will be able to see the names of the amino acid residues. So when we have it in this format, and we just ensure that okay, this format clearly shows what really happened at the active site. We just come to this command line right here. Then we say ray. We input the command ray. Then we go for the resolution that we want. If the resolution that you want is maybe 360, 1080, 420, and so on, you input it. So I will just go for 360 because the higher the resolution, the longer the time it will take. And you click on enter. So when you click on enter, it generates a PNG, which is what we have here. Then you can save the PNG. You have export image as PNG. You save PNG image as. Then you navigate to the folder. Then you can then select. You can then name it. You can then name it. So although we may not be able to fully get what really happened here, but it's something simple, and maybe I will make a separate video to illustrate that better when I have the amino acid residues that really participated in the active site. So moving on now, we are going to quickly go over. We are going to quickly go over 2D visualization. For 2D visualization, we are going to be using a software called Plot. We are going to be using a software called Plot. So, and you can easily download Plot. Just search and you're going to have it. So you need to have Java installed on your system before you can use Plot. So when you download it, it's going to be in your C panel. So you just navigate to local disk, then you are going to see it there after installation. Then you launch. So when you launch, the complex that you saved, the one that I saved earlier when I navigated to PyMod, then open the structure of the protein, then open the structure of the ligand, is what we are going to use for the visualization as well. So you open this particular software. Okay, when you open, this is the software now. Then you click on files. Okay, so you click on files. You click on open. You are working with a PDB file. You come to PDB. Then you browse. Then you navigate to the folder that contains the complex you are trying to view. This is the folder that contains my complex. So I have to navigate down to this folder. So I'm going to come to this PC. Then the folder is pictures. I'm coming. Open PDB. Then you come to pictures, which is my own folder. So now we are going to come to this folder. Then this is the complex. So you click on open. So when you click on open, it will automatically identify the ligand itself. It will automatically identify the ligand that is present in the active site of the protein itself. So we can see right here it's saying PDB code contains one protein chain, which is the chain A, and one ligand. So you are going to click on run. So when you run, you are going to see the 2D visualization. You are going to see all the amino acid participating in the. You are going to see all the amino acids that are participating in the interaction. So this is it now. Now this that we have here is our ligand, and these are the amino acid residues of the protein that are interacting with the ligand. We can see this is an hydrogen bond. This is another hydrogen bond. Cysteine 6913 is forming hydrogen bond with our ligand. We have another hydrogen bond here, which is glycine 6871, also forming hydrogen bond with our ligand. So these ones, the interaction is it is a non-bonded interaction. It could be hydrophobic or any other type of interaction. So which is what we have here. They are non-bonded, but other ones like this that it's forming a kind of connection. This green line that appears broken, it's signifying hydrogen bond interaction. This one here is also signifying hydrogen bond interaction. So we can then save this. We can just click on. We can use sniping tool on our system. You can just use sniping to Windows and Shift and S. Windows, Shift and S. Then you can easily save this. Coming. Just click on Windows, Shift and S. Then you can easily save this. So now, someone was asking earlier how to define the amino acid residues that are binding, the amino acid residues that are interacting with the cognate ligand of a protein. This is the same manner for doing that. So let's say you want to start your docking freshly now. I'll quickly go over that after saving our 2-day interaction. Come my system can be a bit slow. So just
Victor,
yes, sir. I'm almost done.
Victor.
I can hear you, sir. I can hear you,
Yeah,
sir.
once we are done with this, I think we
Hello, sir. Hello, sir. Can everyone hear me? Hello.
I can hear you.
Okay, sir. So I'll
Taking
just
time.
yes, I'm almost done. In 2 minutes, I'll be done. I just want to explain something that when you download a protein from a PDB that has a cognate ligand, and you want to define the active site of the protein, you can use this same software to define the active site, just like you can use a Discovery Studio. You can use this same software. So you just click on File, you load that protein, in the same manner. You load the protein, let's say 3JZK now. So when you download the protein and you import it just like we did, you are going to see an interface like this. So when you see an interface like this and you click on one, it's going to help you identify all the amino acid residues that are interacting with the protein that you downloaded, all the amino acid residues interacting with the cognate ligand. So in this way, you can then manually write this down and then take it for molecular docking. So if there's any question on that, we can ask later on. But this is one of the ways to get the amino acid in the active site of the protein we are working with. If the protein has a cognate ligand, I think I will be handing over now. If there's any question, you can please ask the question.
thank you, Victor. Thank you, Victor, for your presentation. I hope we all understood the workflow it took us through. So please, any question before we go? Just we will only entertain 2 questions. So tomorrow we'll come back and we attend to questions. So I think there's no question. So we'll call the session a day, and we'll continue tomorrow same time. So tomorrow, what is remaining today is the admit profiling, and tomorrow you will go through the 3D interaction again using the amino acid residue that we should have used today. So I will say I appreciate your coming for this free training. I know your time is very important, but at least we just have to squeeze our time and do this. So I thought we would be able to finish it today, but unfortunately we have to continue tomorrow. So I'll try and make time tomorrow, but same time, by the special grace of Allah, I will continue with the remaining part of the training. So thank you very much, everybody. Thank you for Chris. Thank you, Victor, and every other participant. I wish you guys a good day.